Dear ladies and gentlemen, today I would like to discuss on cellular mechanism and iron regulation in cystic fibrosis. I am Mohsina Mukti, studying in the Faculty of Pharmaceutical, Biomedical and Veterinary Science at University of Antwerp. Cystic fibrosis is a lethal autosomal recessive genetic disorder. Only 70 to 75% of gene mutations are responsible for CF have been discovered. The gene responsible for the disease is located on autosome 7. Genes are recessive and occurs only when affected homozygous recessive genes are present. It has highest prevalence amongst the Caucasians, even though all races are affected, which is over 70,000 children and adults. The frequency is 1 in 29 people, and average life expectancy is 31 in male and 28 in female. You can see in the picture that it affects a lot of organs, good number of organs, which includes lungs, where infection and inflammation occurs. It includes pancreas, liver, intestine, sweat glands. It also causes congenital malfunction of vas deferens in male reproductive organ, causing male infertility. Cystic fibrosis is a disease which is mainly caused by deregulation of ions across the cell membrane. It is mainly caused by disease gene of CFTR, cystic fibrosis transmembrane conductance regulator. The CFTR regulates epithelium chloride ion. This CFTR channel also influences other ion channels such as epithelial sodium channel. The function of ion channels in epithelium, besides the general function of regulating osmolarity, and ion channels such as CFTR provides more specific function. By pumping chloride ions out of the cell, CFTR can regulate water secretion in some epithelial cells, for example in the pancreas and intestine. By pumping chloride ions into the cell, the CFTR can regulate absorption of ions in excess of water, thereby creating hypotonic water condition outside the cell. Before we understand the mutations of CFTR protein, we need to understand the normal structure of the CFTR protein. There are 3 types of domain in CFTR protein. The first one is the membrane spanning domain, MSD. The 2 set of 6 membrane spanning segments that anchor the protein to the to the plasma membrane and form ion channels. It also represents 19% of the total protein. The second one is the nucleotide binding domain, NBD1 and NBD2, which is responsible for binding and hydrolyzing ATP. It also controls ion channel gating. The third one is the regulatory domain, which is responsible for activation of CFTR. There are various classes of mutations which ultimately results in the impairment of CFTR function. You can see the class 1 mutation occurring inside the cell membrane. It is ultimately caused by the premature termination codon, and which results in truncated proteins. There are also splicing abnormalities occurring from unstable mRNAs. This leads to severe reduction in CFTR production. Roughly about 5% of the total mutations fall into class 1. The class 2 mutation, as you can see, it occurs in the cell cytoplasm outside the nucleus. This mutation includes common DF508 and deletion of PHE codon, which results in CFTR that is not folded properly. Therefore, protein is degraded in the endoplasmic reticulum, and it represents large majority of total mutation, which is 75%. You can also see in the picture the class 3 mutation, which is occurring in the nucleotide binding region. It also affects the regulatory domain, and the class 4 mutation, which is occurring in the membrane spanning region and does not allow the chloride ion to exhibit its function properly. You can also see the mutation of class 5 occurring inside the nucleus and class 6 in the cytoplasm. The ultimate consequence of a defected protein results to drastic change in the lungs. Only 50% of the synthesized CFTR is correctly folded and trafficked to the cell surface membrane to carry out its function. So as you can see in the picture, this is a respiratory tract where you can see the thick mucous membrane being formed, and which results in persistent bacterial infection by Pseudomonas aeruginosa, Burkholderia cepacia, and Staphylococcus aureus. There is insufficient clearance of bacteria, and lung cells are ultimately destroyed by the immune cells, for example, neutrophil. There are certain current treatments available, for example, treatment by antibiotics such as tobramycin. There are smooth muscle relaxants such as albuterol, and anti-inflammatories such as ibuprofen. There are also DNase, deoxyspiraguline, which affects the internal organelles of the cell. There are also other antibiotics such as gentamicin and CPX, which is an adenosine receptor antagonist. So finally, we reach here to summarize about what we have learned. Thank you everybody for listening to my presentation. Hope to see you again.